Hello everyone. I want to start by asking you to imagine something. Picture yourself waking up on a fine morning, picking up your toothbrush, but you drop it in the sink. You go to tie your shoes, but the laces slip right out of your fingers. Confused, you visit the doctor and they assure you it's just a lack of sleep. But the symptoms worsen. Another visit to the doctor, more tests, and you hit with a life altering diagnosis: amyotrophic lateral sclerosis or ALS. My name is Laiba Imran, and along with my team, we have dedicated our research to finding potential treatment options for this cruel disease. ALS is a progressive neurodegenerative disorder that affects motor neurons in the brain and spinal cord, impacting millions, including famous individuals like Stephen Hawking and the legend baseball player Lou Gehrig. And yet, we know so little about its origins, and worse, there is no cure. ALS exists in two forms: sporadic, which is non inherited, and familial, which is inherited. Combined, ALS affects 2 to 3 people per 100,000 annually, with an average survival time of just 3 to 5 years post diagnosis. The disease starts with mild symptoms like weakness and progresses to total paralysis. Currently, there is no cure. The available treatments only slow the progression of the disease. Therefore, we decided to focus our research on the genetic aspects of ALS. Our research objectives were clear: identify differentially expressed genes, perform gene ontology, and investigate drug gene interactions to repurpose ALS drugs. Here is the workflow we followed. We began by hunting for genomic data from public databases, then proceeded with data preprocessing and normalization. We identified DEGs and finally performed gene ontology and further drug interaction studies. Coming on to our results, we started by collecting data from the Gene Expression Omnibus, focusing on a dataset by Dancon et al. We then preprocessed and normalized the data to identify differentially expressed genes. In total, we found 7,031 genes for familial ALS and 7,044 for sporadic ALS. These were further filtered based on p-values and log-FC change, narrowing down the list to 78 DEGs in familial and 130 in sporadic, which were highly specific and significant. Here are the top 5 most significant genes from both types, including genes like EQF1 and AXIN1 for sporadic and CFDM52 for familial ALS. The volcano plots labeled in red and blue are basically responsible for this deadly disease. We then conducted gene ontology analysis to categorize genes based on their biological processes, cellular components, and molecular functions. This analysis helped us understand the role of these genes in ALS. To better understand how these genes interact, we used Cytoscape for network analysis. Here, you can see the network of 132 sporadic ALS genes with an interaction score of 5, showing how they are beautifully connected, and a similar network for the 78 familial ALS genes with an interaction score of 4. Next, we explored drug repurposing using DGIDB. From our analysis, we found 752 drugs for sporadic and 609 for familial ALS. We narrowed these down to 8 sporadic and 7 familial drugs based on their interaction score and FDA approval. After cross-referencing with DrugBank, we proposed a combination therapy of 4 drugs that we believe could be highly effective in targeting the specific molecules responsible for ALS. Finally, we visualized the proteins produced by these genes. These are the 3D structures of the proteins responsible for ALS. This allowed us to better understand the functional impact of the mutated proteins. In conclusion, our study identified a significant number of DEGs related to ALS. By targeting these genes through personalized medicine, we can improve prognosis and potentially extend the survival of ALS patients. For future work, we aim to take this research into the laboratory, conducting in vivo experiments to validate the efficacy of our proposed drugs. These are our references. Thank you very much.